How are we doing, guys? It's Mr. Striz. I'm checking in. Today's LEQ is how does transcription and translation occur? So we talked about DNA replication, and now we're going to go 2 steps further in how proteins are actually made from that replicated DNA. So the first thing we're going to talk about is transcription. So as you notice this picture here, you have the DNA. We are now introducing RNA in this transcription process here, and then translation, which we'll get to in a little bit later on. But what transcription is, it reads the base sequences on the genes of DNA and converts them into mRNA. So notice on this picture here, this is what we talked about already. We had that strand of DNA and we replicate that strand of DNA. Now during transcription, RNA polymerase will come in and unzip that DNA strand, and then notice it's going to add an RNA sequence there. So now it's kind of like replicating again, but with RNA instead of DNA, and that occurs during transcription, and it makes a whole new sequence there. Now, you need to know where transcription takes place, and it's the same place as DNA replication, but it occurs in the nucleus. So transcription occurs in the nucleus. As you'll see, eventually it's going to move out into the nucleus and into the cell. I brought up RNA polymerase just a little bit ago, and we'll get into what that is. So notice here, RNA polymerase is an enzyme that opens up the replicated DNA strand and uses it as a template for RNA. So as you see here in this picture, this RNA strand is being made. Well, what's occurring is that RNA polymerase, that enzyme, is using it as a template. So now we have this RNA polymerase creating this new strand of RNA. And it's similar to DNA polymerase, because remember, DNA polymerase, we talked about in the last video, where it makes a new strand of DNA. This one is just doing RNA. Now, moving on to that translation part. So we have this transcripted RNA sequence right here that we got from this DNA sequence, and now we're moving into translation. So what translation is, its purpose is to translate mRNA to produce a specific amino acid, which later becomes a protein. So if you go back to that video there, or back to that picture a little bit ago, in translation you have this mRNA, and now this mRNA, it's broken up into codons, or these 3 nucleotides, which we'll talk about that later again, but it now becomes into amino acids, and those amino acids form a protein. So you break those up, like that AUG, it stands for MET, the amino acid MET. It's like the start amino acid there. And so they all code for different amino acids in translation. So transcription, DNA is made into RNA, and then in translation, RNA is then put into an amino acid, which is then made into a protein. So once again, it translates mRNA to produce a specific amino acid, which later becomes a protein. Now, where does translation take place? So we talked about already where transcription takes place. We talked about transcription taking place in the nucleus. Now translation occurs in the ribosomes. And that's the reason why is because we talked about ribosomes back when we talked about the cells. Remember, ribosomes is protein synthesis. So proteins are made in ribosomes. That's where translation occurs. Now, moving on to the next question there, what are proteins made of? Proteins are made of amino acids. That goes back to our macromolecules, where the amino acid is the monomer of proteins, because all the amino acids form a protein. Now, what does each codon represent? Now, I talked about already how in translation it's broken down into different codons. Notice a codon is a set of 3 nucleotides there. Now, the codon is a sequence of 3 nucleotide bases that goes with a specific amino acid. So you have the codon GCU. Now notice this is a codon table, and we'll take care of that another day. But notice if you go to GCU codes for alanine. So that GCU is a codon that codes for the amino acid alanine. Now let's just take another look. ACG, go with the A, C, and find the G. ACG right there. ACG right there, translate to that protein, that amino acid, and then those eventually become proteins. So codons are just a sequence of 3 nucleotide bases that go with a specific amino acid. Now we're going to continue on with the video, but we're actually going to go onto the left-hand side there. Those 4 questions that you see there from the video, Alright, we're going to talk about first RNA. The function of RNA converts information stored in DNA into proteins. So RNA takes that information that DNA has and then takes that information and then converts them into proteins. And that's through the process of transcription and translation. Now, what about mRNA? mRNA makes a copy of DNA and carries it to the cytoplasm. So we saw again in that picture where that mRNA made a whole nother sequence from that replicated DNA strand, and then it carries that into the cytoplasm. tRNA decodes mRNA to make it into a protein. So the tRNA will take that mRNA sequence and then it breaks it up into those codons, and then those codons make amino acids, and amino acids are turned into proteins. And then finally, what are the base pairs for RNA? Okay, so if you notice here in transcription and translation, with RNA, the base pairs for RNA is a little different than DNA. So adenine bonds to uracil. So A is actually bonding to U now. A no longer bonds to T. That only occurs in DNA replication. Now thymine still bonds to adenine, so T to A. And then cytosine and guanine still bond to each other. So cytosine bonds to guanine, or guanine bonds to cytosine. So C to G or G to C. So remember, the big difference between RNA and DNA, adenine bonds to thymine, the A to T, but with RNA, adenine bonds to uracil, or the U. And you actually have some practice underneath there that you'll go through with the video as well. But now I have a little video itself on transcription translation that we'll watch to end this video here.
Gene is switched on. An enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process...
So notice right here you have an A to U. So just remember that adenine bonds to uracil in RNA.
This is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.